These are the second set of photosynthesis notes on the light reactions. Light is a form of energy. It is a form of radiation. The sun emits all kinds of radiation, but there is a narrow band of energy on the electromagnetic spectrum that is called visible light. All life depends on this very narrow band of radiation called visible light. Light behaves like a wave, and these wavelengths of visible light are the wavelengths that are important to life. These wavelengths typically vary from 380 nanometers to 750 nanometers. You do need to know the order of the wavelengths. Roy G. Biv is the easiest way to remember it. Red, orange, yellow, green, blue, indigo, violet. However, even though we usually use the acronym Roy G. Biv, they do go in reverse order on the electromagnetic spectrum, where red would then be the longest wavelength and purple would be the shortest wavelength. This particular graph is called an action spectrum. An action spectrum is an important graph for photosynthesis because it shows you how much photosynthesis, the rate of photosynthesis that's happening at each different wavelength of light. Light also behaves as if it consists of these little bundles of energy that we call photons. The amount of energy in one photon is inversely proportional to the length of the wave. So although red is the longest wavelength, it would have the least amount of energy per photon. Violet is the shortest wavelength. It has the most amount of energy per photon. Typically, for plants, they absorb the best in two areas of the visible spectrum. It is usually in the areas of blue and red light energy. And this is because of the pigments that they tend to have. Chlorophyll A is the main photosynthetic pigment, so all photosynthetic plants will have chlorophyll A. And so this is where chlorophyll A actually absorbs the best. And you can see that on this absorption spectrum. This is a little bit different from the last graph. The last graph was an action spectrum. Action spectrums graph the rate of photosynthesis on the y-axis, but the absorption spectrum graphs the absorption, the amount of absorption that's occurring on the y-axis. The x-axis is the same. It will always graph the wavelength. Oops, sorry, going back. And this is specifically for chlorophyll A. So if we looked at a different pigment, then the graph shape would look slightly different. On top of that, you should notice that the absorption and action spectrums do look similar in shape. This is because if the plant is absorbing the best in these two areas, then they would be doing the most photosynthesis at those two wavelengths as well. So there is definitely a correlation there. This graph shows several different pigments. So notice that here's chlorophyll A, the blue line, the same two peaks we just talked about. And then here is chlorophyll B, which is just slightly shifted over. And then there's other pigments here as well. These other pigments typically act as helpers or play other roles for the plant, but they are not the main photosynthetic pigment. We'll come back to chlorophyll A in just a bit. That one's always the main photosynthetic pigment. Here's a note that you do need to make note of. IB wants, when you graph action or absorption spectrums, that you graph both the wavelength and the color of light on the x-axis scale. So notice that we do still have our wavelength scale evenly distributed, and then the colors are listed below each of the different areas of the visible spectrum. This is because we do mostly associate colors with visible light. However, it's the actual wavelength that is being absorbed. And this is the only graph on the notes that does show both on the x-axis. Plants have pigments, lots of different kinds of pigments. Chlorophyll A is the main photosynthetic pigment. Chlorophyll A looks like this, with this R group being a methyl group. So this R group right here. Chlorophyll B is a very similar molecule. It also looks like this, but this R group is slightly different. This means they have different functions. Structure leads to functions. The job of chlorophyll B is to typically pass photons of energy to chlorophyll A. It's like the helper in photosynthesis. Other pigments can also be considered these helpers as well. Any pigment that's helping chlorophyll A, we call that an accessory pigment. Carotenoids are other types of pigments. These carotenoids typically show as yellow or orange in a plant, whereas chlorophylls are the reason why plants are green. They are not using green light, they reflect it back, therefore plants are green. But sometimes you'll notice at certain times of the year, the leaves will change color, and then you'll start to see these yellows and oranges. We'll talk about that more in just a second. Carotenoids in general are thought to play a role in what's called photoprotection. It means that as those photons of light are coming in, that the carotenoid pigments will kind of dissipate back out the excess light. This helps to protect the plant from getting too much light. Here we can see those different colors. We have our reds and oranges and yellows. Typically yellows and oranges are due to those carotenoid pigments, but the reds are due to a different pigment called anthocyanins. These pigments are always present in the plants, however, we don't typically see them until the fall when the leaves change color. This is because during this time when the temperature starts to decline, chlorophylls will start to break down. So once the plant starts to break down its chlorophyll pigments, we can now see these other pigments shining through more. They were always there. They were just masked by the really high levels of chlorophylls that are present during the warm temperature months, since chlorophylls play such a big role in photosynthesis. This picture is showing you that as the light energy comes in as a photon, it's going to allow for us to take electrons that are considered to be ground state electrons, which means they don't really have a lot of energy, and receive that energy from the sun. This puts those ground state electrons now into a more excited state. This excited state is going to be important because we are going to be passing those electrons to electron shuttles, and electron shuttles are going to carry high energy or excited electrons to another location. Here just shows you the same kind of image, however, where those ground state electrons are going to be is going to be in chlorophyll A. So we're going to talk more about that. This just kind of associates the electrons with the pigments. In the chloroplast, we are going to have a molecule right here that is called a photosystem. The photosystem sits in the thylakoid membranes. So here shows you, here's a thylakoid disc, here's the thylakoid membrane. These photosystems are going to be a very important part of the light reactions. When we look at what's happening here, you can see that we have chlorophyll A right here. This chlorophyll A is special. It's an association of two chlorophyll A molecules. It sits in an area of the photosystem that we call the reaction center. So this light purple portion of the molecule is the reaction center. It's still part of the whole photosystem, but this is the reaction center. Also in the reaction center is a molecule called the primary electron acceptor. What we're going to see is chlorophyll A in the reaction center holding those ground state electrons. Photons of light energy are going to come in. That energy is going to be passed from from pigment to pigment to pigment to pigment to pigment until eventually that energy is received from chlorophyll to chlorophyll A in the reaction center. This is how our electrons will move from ground state up to an excited state, and they will be received by the primary electron acceptor. A photosystem that we just looked at on the last slide is considered to be a molecular array of chlorophyll and accessory pigments. You saw the chlorophyll in the reaction center. The accessory pigments are all the other pigments outside the reaction center. Let's go back and look. So these are considered the accessory pigments. They can be chlorophyll Bs, they could be other types of pigments, but their job is to pass energy to the special chlorophyll A's here in the reaction center. They could even maybe be other chlorophyll A molecules, but notice these are single pigments. The pigments inside the reaction center are special. There are two different photosystems that will be embedded within the thylakoid membrane that we'll talk about, photosystem 1 and photosystem 2. They absorb in just slightly different areas of the visible spectrum, and this allows for them to have less competition of light with each other because they will collectively work together. Even though they're called photosystem 1 and photosystem 2, when we look at the overall chain of molecules that's used in the light reactions, photosystem 2 actually comes first, and then photosystem 1. This is because they were named in order of discovery, not in the order of their usage. So again, here shows you a photosystem, and here's those special chlorophyll A molecules in the reaction center. Here's our accessory pigments. They have to work collectively together. Neither of them can work on their own. Again, they do have to work together to be able to perform the reactions of photosynthesis. This is just a different view of a photosystem. It's a different drawing. But again, light energy coming in, passing from pigment to pigment, eventually passing to the chlorophyll A's of the reaction center. Once that energy is received, the electrons move from ground state up to an excited state, and they will be passed to the primary electron acceptor. Here shows you where we're going next. So here's photosystem 2, first in the membrane, then photosystem 1. Water is going to be responsible for our electrons going to photosystem 2. Water doesn't contain a lot of energy, so when the electrons are given to chlorophyll A, they are ground state. That's why they're ground state. Photons of light come in, pass that energy to chlorophyll A. Those electrons move from ground state up to an excited state. They are received by the primary electron acceptor. Now these high energy electrons are going to go down an electron transport chain. They're going to get back to photosystem 1. They're going to get re-excited and go down a second electron transport chain, and ultimately they can leave in an electron shuttle. We're also going to make some ATP. So we'll talk about this in more detail. But notice that they work together. So being P680 and P700 and allowing them to absorb in just slightly different areas of the visible spectrum leads to less competition of light between them. That way they can work better together more efficiently. So we're looking at a picture in a eukaryote because we're in the thylakoid membrane in a chloroplast. But remember, prokaryotes don't have organelles. So where would you think that these particular molecules are going to be located in photosynthetic bacteria? They would be located in their cell membranes. So they still do all the same reactions, they just don't have organelles. They have to have the molecules for both photosynthesis and cellular respiration embedded within their cell membrane. Here shows you the stacks of granum that are all together here, and look at how many of these electron transport chains and photosystems are embedded all throughout this. This, remember, means that that's a lot of surface area. Structure leads to function. We can do more light reactions because of all this membrane. Light energy is going to allow for us to make ATP and NADPH in the light reactions by using the photosystems. However, this transformation of energy can occur by two different ways. So it can go via cyclic electron flow or non-cyclic electron flow. We'll do non-cyclic first, and then we'll do cyclic. In non-cyclic electron flow, water splits. This is called photolysis, and it gives its electrons to chlorophyll A in the reaction center. This is where oxygen is given off as a waste product. So again, water splits, gives its electrons to chlorophyll A. These electrons then become excited from the sun's energy and gets passed up to the primary electron acceptor. Once received by the primary electron acceptor, it's now ready to go down an electron transport chain because it has enough energy to do so. Again, this is called photolysis. As soon as the electron moves up to the primary electron acceptor, this reaction will happen immediately again, filling the hole of chlorophyll A. This is because chlorophyll A is so electronegative, it's not going to sit empty. It's going to want to have those electrons all the time. So this reaction just keeps happening over and over and over again. Remember that even though oxygen is considered a waste product of photosynthesis in general, being able to have the process of photosynthesis happen a lot more way back in evolutionary history has allowed for immense changes to occur on Earth. Incredible consequences for living organisms as well as geological processes on Earth. So this process, and it's increasing in its ability to occur, is incredibly important. So again, water splits, gives electrons to chlorophyll A. This is where you see oxygen leave as a waste product in photosystem 2. You will then have the electrons move up to an excited state, received by the primary electron acceptor, and now they will flow down an electron transport chain. You don't need to name the molecules, you just need to know that they go down an ETC. There are 3 proteins of this ETC, and then ultimately those electrons are going to get passed to photosystem 1, it's chlorophyll A. Because we are passing electrons, notice one thing that we're doing is pumping hydrogen ions. Okay, so this is going to be very similar to the process we talked about in cellular respiration. In fact, this is also chemiosmosis. So hydrogen ions are going to be pumped from high concentration in the stroma to low concentration in the lumen. Oops, back up. From low concentration, in the stroma to high concentration in the lumen, low to high. So energy will be needed to activate the pumps. Technically, only one is a pump. So we're going to pass the electrons here, and they're going to have the most energy here. Then we're going to pass those electrons here, and each time the electron is passed to the more electronegative protein in the chain, that electron will drop in energy as we pay to pump the hydrogens into the lumen. Hydrogens are pumped from low to high concentration into the lumen. Eventually, this electron makes its way here to photosystem 1, and it's back at ground state. But in the meantime, proton motive force is building up in the lumen. Once there is enough proton motive force that builds up, it will cause the shape change of ATP synthase enzyme, powering the production of ATP in the stroma. This ATP will be used in the stroma to power the Calvin cycle. This is often called non-cyclic photophosphorylation because it's occurring during non-cyclic electron flow, and light energy ultimately helped to drive this process of phosphorylation of ADPs into ATPs. We'll pick up right back here on a future slide. So again, water splits, gives its electrons to chlorophyll A. It becomes excited from the sun's energy, passes down an ETC. This ETC pumps hydrogens from low concentration in the stroma to high concentration in the lumen. Once there's enough of that, a shape change of ATP synthase will occur. Proton motive force will allow for this rush of hydrogen ions into the stroma, powering the production of ATP. We'll talk about this in a second. Water splits, donates electrons, moves up to an excited state, passes down an ETC, hydrogens are pumped, proton motive force builds up in the lumen through chemiosmosis, power the production of ATP in the stroma. This ATP is made in the stroma and will be used in the stroma to power the Calvin cycle. So remember, eventually the electron makes it to photosystem 1, it's back at ground state. So now we're going to talk about what happens here. Since it's back at ground state, going back, it needs an additional bump of energy from the sun. This is why it's important for no competition between the two photosystems. They will move from ground state to an excited state, and they're going to pass on a smaller ETC. This ETC is only one protein. This ETC is responsible for passing the high energy electrons to an enzyme that's called NADP plus reductase, which reduces NADP plus into NADPH. Now we've made a reduced shuttle. This reduced shuttle also goes to the Calvin cycle to help power the Calvin cycle. And here's the word forms of that. So again, when electrons reach the end of the ETC, they make it to photosystem 1, they become excited again, they go down another ETC, which is much smaller, this one, and that passes to an enzyme that's called NADP plus reductase enzyme. The job of NADP plus reductase enzyme is to attach electrons to NADP pluses, the oxidized shuttle, forming the reduced shuttle of NADPH. NADPH then goes to the Calvin cycle. So here it is again, whole picture. Water splits, donates electrons to chlorophyll A, a pair of electrons. Oxygen leaves as a waste product. Light comes in, energies pass from accessory pigment to pigment to pigment, till eventually it's received by chlorophyll A, moves from ground state to an excited state, is received by the primary electron acceptor, passes down an ETC. This ETC is responsible for pumping hydrogen ions into the lumen, generating the proton motive force that lets us make ATP through chemiosmosis. Eventually, the electron makes it to photosystem 1's chlorophyll A's. It's back at ground state because we paid to pump the hydrogens. Light energy, again, excites these electrons, moves up to the primary electron acceptor, passes down a second ETC, which is much smaller, just one protein. Those electrons then get passed to an enzyme, NADP plus reductase, which reduces NADP plus into NADPH. All products are going to the Calvin cycle, all energy products. NADPH and ATP go to the Calvin cycle. Oxygen, waste product, does not go to the Calvin cycle. Okay, so here it is again. You can see water splitting, photolysis, excitement of those electrons, moves down the ETC, hydrogens are pumped into the lumen. Once proton motive force builds up, shape change of ATP synthase. Now we've made ATP to go to the Calvin cycle. The electron is back at ground state since we paid to pump the hydrogens. They have to get re-excited, goes down a second ETC, passes to NADP plus reductase enzyme, which reduces our shuttle. The shuttle also goes to the Calvin cycle in the stroma. What's nice about this picture is that you can see that after the Calvin cycle uses the ATP made in the light reactions, it sends back the empty ADPs to keep getting refilled. That's how there's always like enough ADPs to refill. This one shows the same thing with the shuttle. NADPHs drop off, they get oxidized in the Calvin cycle, and then they get returned as their oxidized versions, NADP plus, to the light reactions so that we can keep refilling it. These two reactions do depend on each other. The Calvin cycle is where we're going to use our CO2 and our energy molecules to make a sugar. We've talked about adaptations of the mitochondria, and the chloroplast is similar. It has a lot of surface area of the thylakoid membranes. This allows for more electron transport chains, which means more light reactions, which means more energy molecules to the Calvin cycle, which ultimately means more glucose to the plant. That's important. Another important adaptation is the small volume of the lumen. This allows for proton motive force to build up quickly. leading to more ATP production. This is very similar to how we said the intermembrane space of the mitochondria had that small volume as well, which did the same function. And then lastly, compartmentalization of enzymes and the substrates that are used in the Calvin cycle. This means that it keeps everything all tightly in the stroma rather than floating around in a much bigger space, such as the cytoplasm. So having all of that in one place, the stroma, allows for their concentration to be higher, and this allows for the reactions to occur more often. Again, very similar to the enzymes of the Krebs cycle and the link reaction being compartmentalized in the matrix of the mitochondria. In cyclic electron flow, it's going to be a little different. Only photosystem 1 will be used, so we won't use photosystem 2. We're only going to make ATP. We're not going to make any NADPH or release any new oxygen. So, cyclic electron flow is considered to start here in photosystem 1. It's considered to start once it gets to chlorophyll A of photosystem 1 and it's back at ground state. This is really where the electron is going to have a choice to be made, whether it's going to go cyclic or non-cyclic electron flow. So it's going to become excited from energy from the sun, and it's going to go to the first or the second ETC. Once it's in the ETC, a choice has to be made. If NADP plus is available, like if it got shipped back by the Calvin cycle, then the electron will go non-cyclic electron flow. However, sometimes that energy shuttle is not there to fill up. If it's not there to fill up, it cannot be passed to the enzyme NADP plus reductase. So you can see it's crossed out. No, it is not going to take those electrons because there is no empty shuttle there yet to fill up. So then that little protein of the second ETC actually moves backwards in the membrane of the thylakoid and drops off in the first ETC. So then the electron, this high energy electron, is going to go down our first electron transport chain, and eventually it's going to come right back to the same chlorophyll A molecule it left. Therefore, it's a cycle. But because it does use this first ETC, it will pump hydrogen ions into the lumen. Therefore, it will contribute to proton motive force, and it will contribute to the production of ATP. So it's a cycle because it starts off in chlorophyll A of photosystem 1 and cycles all the way right back to it. We never use photosystem 2 at all. It gets bypassed, it gets dropped off after that. So therefore, no new oxygen is evolved in this process. And we don't make a shuttle since there's no empty shuttle to fill. So here is that picture. Again, cyclic electron flow starts here. After it goes down the ETC, the electrons are going to be here in chlorophyll A. They become excited. It will pass to this protein of the second electron transport chain. It's called FD. You don't have to name it. I usually call it the carrier protein because it's mobile. And I know it's grayed out here, but that's because it's already showing that it has moved over here. So it will go up and pass to this molecule. It will try to pass to NADP plus reductase enzyme, but when there is no NADP plus available, it cannot pass. If it can pass, it would go non-cyclic. But since it cannot pass, we need to keep the molecules moving. Therefore, this protein moves backwards in the chain and drops off in the first ETC. Therefore, it will use the first ETC to pump hydrogens, leading to proton motive force, leading to ATP production. Cyclic electron flow. It's like a backup plan. Once it gets back here, it will once again try to go non-cyclic. If it can, it will. If it cannot, cycle back. The preferred pathway is always non-cyclic because you make more energy. You make NADPHs and ATPs, but we don't want to get a blockage of the chain, so it needs to be able to have an alternative pathway to keep moving. And it does end up being a really good thing for the plant anyways, because when we do the Calvin cycle notes, you will see that the Calvin cycle actually consumes more ATP than it does NADPH. So it also allows for us to make enough ATP compared to NADPH to drive the Calvin cycle. So it is this kind of balance for the cell. And that's it for the notes part 2.